My name is Awella Onwubuya. I am a student at Liberty University in the Masters of Arts in Biomedical Sciences program, and this is a presentation on the ergogenic effects of caffeine. So, to understand the ergogenic effects of caffeine, you would need to understand three different components, and one of these components is the motor unit. And the motor unit can be defined as the muscle fibers in which are innervated by a singular motor neuron. Longitudinal columns of motor neuron cell bodies are arranged in a nuclei, and innervation by a singular motor neuron is done by a single motor nucleus. Another component you would need to understand is motor unit recruitment, and this is the sequential activation of the same or supplementary motor units with an increase in strength of intentional or voluntary muscle contractions. And the central nervous system has an impact on muscle recruitment. It increases number of active motor neurons. It also increases the firing rate of a singular motor unit to optimize the aggregation or the summation of tension gathered. And then finally, you have the EMG, which is a technique used to understand patterns of production force exerted by skeletal muscles. And it records motor unit activity by placing electrodes on the muscle. The electrodes are then connected to an electromyography. Then the recordings of the muscular activity is called an EMG. So in the experiment which was observed, which was an aerobic or endurance activity, your body would naturally use glycogen as its energy source. But with the addition of caffeine, cyclic AMP phosphodiesterase, which converts CAMP to 5-AMP, was blocked. And this results in glucagon action being enhanced, which would result in longer durations of CAMP levels. Glycogen would then retain its phosphorylated phase for longer periods, and glycogen would remain upregulated for longer periods. Caffeine is a stimulant, so additional amounts of glucose would remain in the bloodstream for longer periods of time. So understanding the motor unit and the motor unit recruitment and caffeine's ability to enhance glycogen activity within the bloodstream, my hypothesis, a reasonable hypothesis to me, would be that because caffeine reduces the depletion of glycogen by inducing the body's use of adipose as fuel, the effects of caffeine will aid in the conservation of energy over longer periods of time. And we're speaking about aerobic or endurance activities, which was seen in this experiment. So the materials used to carry out this experiment were a 15-pound dumbbell, 5 shielded lead wires, bioamp cable, disposable electrodes, dry earth strap, PowerLab 26, the lab chart software, caffeine tablets, specifically 200 milligrams, glucose tablets, specifically 4 grams, and sugar water, specifically 19 grams. And the procedures to carry out this experiment where the subject would take the 5 shielded lead wires and attach them to their dominant arm. You would do this by placing 2 lead wires on the superior portion of your bicep and 2 more lead wires on the inferior portion of your bicep region, and this is on the region of your arm. And you would place 2 other lead wires on the superior and inferior ends of the tricep region of your arm. Then you would place the last lead wire on the wrist of your forearm. You would double check that the lab chart is set up properly before the initial recording. Without any abdominal support, you would use your dominant hand to hold the 15-pound dumbbell at a 90-degree angle. After, you would press start on the lab chart immediately after picking up the dumbbell. You would repeat the methods for your non-dominant arm. So if you were right-handed and that is your dominant arm, you would switch to your left hand. Then you would repeat the entire process every 30 minutes until 90 minutes has passed. 4 conditions were observed during this procedure, and one was baseline, which is no caffeine or glucose. Another one was with caffeine. Another one was with just glucose, and the other one was caffeine and glucose in combination. So the results varied, and I compared 8 different graphs. The first graph was 30-minute baseline versus 30-minute glucose, 30-minute baseline versus 30-minute caffeine, 30-minute glucose versus 30-minute caffeine, and then 60-minute baseline versus 60-minute glucose, 60-minute baseline versus 60-minute caffeine, 60-minute glucose versus 60-minute caffeine, and 90-minute baseline versus 90-minute glucose. And in the results, you can see that in the 30-minute baseline versus 30-minute glucose, the baseline actually recruited less muscle fibers than the glucose. And the same in the baseline versus the 30-minute caffeine. The baseline was seen to recruit less than the caffeine. But when you look at 30-minute caffeine versus the 30-minute glucose, you can see that the glucose actually recruited more muscle fibers than the caffeine did. And the 60-minute baseline versus the 60-minute glucose is similar to the 30-minute baseline versus the 30-minute glucose. And here are graphs of the other 4 comparisons. So you see 60-minute baseline versus the 60-minute caffeine, and you can see that the baseline is recruiting less motor units than the caffeine is. And when you go to the right, you see that 60-minute glucose versus the 60-minute caffeine. You can see that glucose as well is recruiting more muscle fibers than the caffeine would. And in the 90 minutes, 90-minute baseline versus 90-minute glucose, you can see that the baseline is starting to recruit more, but the glucose is still recruiting more muscle fibers. And same goes for the 90-minute baseline versus the 90-minute caffeine. You can see that the baseline is still recruiting less motor units than the caffeine is. So compared to other articles and experiments, our results slightly differed from the recent studies. And in one study, caffeine was only effective in smaller quantities. In another study, there was little evidence to conclude that coffee was even an effective ergogenic aid. And these studies use smaller quantities of caffeine in contrast to the large quantities that we use in our experiment. So maybe that has something to do with deferring results. But caffeine in these studies didn't seem to change much or have much of an effect. So here's a table on the right of the summaries of these studies that utilize caffeine to see if it was an effective ergogenic aid. So, in conclusion, I would, if I had to do this again, I would demonstrate the effects of caffeine maybe in lesser doses, and I would also see the effect that caffeine would have on, different body types, different heights, and possibly different statures, to see if that plays a role in the effect of caffeine. So here's a list of the references I used to complete this project. And this concludes my presentation on the ergogenic effects of caffeine. Thank you.